Now that we've gone through the processes of transcription and translation for gene expression, we can now talk about what happens with mutations. Now, a mutation is a permanent change in the DNA. Mutations can be of a variety of types. They generally result from mistakes in DNA replication. So what we have here is, again, a flow of information diagram where we have the reading frame of DNA, the corresponding messenger RNA, again built according to the base pairing rule, and then the codons and the amino acids that are produced in that are assembled in translation. So if we change something in the DNA, then that change is going to get transcribed into the RNA, and we'll see what happens next. Now, there are different types of mutations. If we simply change a letter for another letter, we call that a substitution or a base pair substitution. Now, what will that do depends on the situation. So, for example, if we were to change this to, say, a G, well, and that becomes a C, well, guess what? UCC still codes for phenylalanine, and this mutation will not result in this protein being different at all. We refer to that as a silent mutation, one that has no effect. Now, also, mutations in non-coding sequences would have no effect because they don't even get transcribed and translated in the first place. Or we could have an amino acid substitution. Well, for example, if we did this, and that becomes this, then we have changed this amino acid to that one. Now our protein has been different. It's now altered by one amino acid. What will that do? It depends. It may change the protein a little bit. It may change the protein drastically. It just depends on which one it's being changed, where it is, and how that affects the subsequent structures of the protein. And that would be referred to as a missense, where it refers to an amino acid substitution, a missense mutation. Now, a nonsense mutation is when a base pair substitution creates a stop codon and stops everything short. So, for example, if we were to change this one, let's see. Well, guess what? We now have a stop codon, and essentially everything else is not even going to be translated at all. We've stopped everything short, and we now have a non-functional protein, a nonsense mutation. So these are the results of base pair substitutions. Now, insertions or deletions. Insertion means you add a new nucleotide. A deletion means you take one away. Now, let's just throw in an insertion and see what happens. Well, let's just put another A here, and that becomes a U here. Well, guess what? We've now shifted every codon over by a letter, and we refer to this as a frame shift mutation. And every amino acid from that point on is going to be potentially different. So these are some examples of point mutations.